47. Okay, yeah. Oh yeah, they're called tagmata. I think I have tagmata in here, right? Okay. All right. All right. So we want to do chapter 33, 47, 40, 41, 44. And then I think that this is all going to be like human stuff. Oh yeah, that's just not going to be on it. 44 will definitely be on it. The immune system would actually be really interesting to learn about. Nutrition, we definitely need to know that. 40, what's in 40? Oh, it doesn't give you the breakdown. Really? That seems weird. Just tells you the chapter. Oh, so this is mostly about homeostasis. Okay. Nutrition. Oh, we got a lot to learn. Alright, so let's just get into it. I already did some of this, so we're not going to get into it, but this is sort of the North Star for understanding this whole thing, is that if you have sort of metazoa, then these aren't really, they don't really have tissues. They don't really undergo gastrulation. So then we have eumetazoa, which does undergo gastrulation. We get Cnidaria, which are diploblastic. And then we get to Bilateria, and Bilateria are triploblastic. And then in here, they sort of call these protostomes, but they're not. I think that's sort of gone by the wayside. But Lophotrochozoa and Ecdysozoa are both protostomes. These Ecdyso means molting, I guess. And then this name, honestly, it's funny because they name it this, but it's really one of these clades that's molecular. Like, they don't really, like, there's a whole bunch of stuff in there. And so anyways, where we're at right now is I did this and I'm at mollusks. So I already learned about sponges. Do I have mesohyle? That's actually a good word. Okay, yeah. Hyle. I don't know what I know what meso means, middle something though, but I don't know what hyle means. Hill? I don't know. Okay, let's just do we have Nito? I guess we had Nito for Cnidarian. What about Nima? Oh, we do. Okay. Okay, yeah, then we have a I feel like this life cycle is relatively easy. What the fuck? Yeah, oh yeah. Lophotrochozoans, a clade identified by molecular data. So I guess memorizing what's in Lophotrochozoa is probably worth doing because there isn't a real anchoring thing to remember that they have. I think the big three are going to be flatworms, mollusks, and annelids. I know they actually break up flatworms into classes or something, but we're not. I don't think I'm gonna do that. All right, just keep moving on. All right, let's just go to where I was. All right, mollusks. So okay, wait. Let me write like 2 sentences about flatworms. So flatworms, gastrovascular, and flat. So excretion and gases from um, diffusion. I mean, is there anything really interesting besides that about I guess maybe we should give some species. Oh yeah, the free living ones secrete ventral mucus and have ciliated ventral side for movement. Also, sometimes, I guess we have sensory and motory mechanisms, but we don't really have anything about like sinusoidal motion or anything. And then parasitic. Include tapeworms. Snails, slugs. Entonces vamos a la máquina. ¿Eh? Okay, I'm gonna try to draw this, I think. I don't think I gave myself enough space. Oh man, this is crazy. What species is this? This is the basic body plan of a... Because they're Lophotrichozoans, they're therefore protostomes. And I guess we definitely need this diagram. Where is it? What the hell? I feel like there should be more. Oh yeah, there is. There's way more. Wait, is this the same? No, E2, yeah. Lectures, lab lectures. E2. E3. And, I don't think SC... Yeah, I don't think that this is going to be on the, No, actually, I forgot. I also have a bio homework due. I don't think we need to do chordates, but bugs for sure. Okay. Okay, so it's probably going to be in, yeah, I have E2, I have E1 open, so open E2, and E3. So, let's see, we're going to need to write all this down, because I need to remember what turns into what. Oh, shoot. Okay, wait, we need it. I should have made a diagram for that. Okay, once I finish mollusks, I'm gonna, return to that and draw this. So we certainly will need to... Auricle? Bro, I don't know what that is. Because I think, you know, it's hard because there's so many sources of information that it's hard to... But we'll definitely need to get into this thing. Oh, wow, I guess he did go into this. But did my book even say Turbolarians? Wait, Annelida is not considered, a flatworm. Flatworms are Lophotrochozoans, but, oh my god, dude, these diagrams are insane. You gotta be an artist to be a biologist, it feels like. Alright, so he's just doing that, but where is mollusk? Mollusk isn't in here? Interesting. Oh yeah, okay, here's mollusk. Triploblastic, bilateral symmetry with cephalization. Except bivalves. And they also, oh yeah, and then gastropods. The mantle cavity is above the mouth. Okay, so he calls them eucoelomate. So they are eucoelomate, but it's reduced, and it's sort of confined to just the heart. It sounds like. Okay, so this is the diagram we just put in here. Polyplacophora. Do I have that? Okay, I do. Okay, yeah, so those are the 4 classes. So, wait, are pill bugs terrestrial? They might be mollusks. No. I thought he said something about terrestrial mollusks that aren't snails. Yeah, it was it was a little cocky of me to only write 3 lines about flatworms and platyhelminthes in general. That is not good. So then, alright, so we'll do... Alright, so chitons is not really that interesting. Chitons are also called Polyplacophora, which is like many plate bearing. Okay. Let's see, do they mention torsion? They don't mention torsion. Wow, yeah, they didn't explain anything about this torsion thing. Okay, I'm good with that. We can keep going with this. We'll get to the bugs, by the way. We're just working our way through the animals, man. Do I really want to draw this? Is this even in the book? Oh yeah, we do have this. Okay. This picture sucks. I don't even know what I'm looking at. How is this the mouth? I don't think I'm gonna write all that down. That's kind of a better picture, actually, than this. This doesn't really show where the food goes. What is the palp? What is a palp? We have so many words to add. Okay, I feel like that's enough on clams. Let's keep going. Bivalves are not that cool. Now, cephalopods are sick. Ooh. Dude, what a garbage picture, man. Come on, dude. That is ridiculous. Is it from... I don't think it's from this... let's just look up. See if we can find a better picture of it. Oh, nice. Okay, so we have a radula. We have jaws. Oh, here we go. Here's the funnel. Esophagus, mantle cavity, gill. Yeah, that makes sense. Ink sac. Right, did he say something about 3 hearts here? Maybe he just said it in person. Or is that the one with 3 hearts? I don't even know. Systemic heart. Where's the 3rd then? Oh, they have 2 gill hearts. Okay. Okay. Oh yeah, this is where it was. Should I try to draw this? This seems like a tough draw. Funnel. Radula. Okay, then this goes into a tiny little esophagus. And then we have a big old brain around this thing. Big old brain. It's interesting that it's like often around the esophagus, no? Okay, and then what is this? There's no arrow to that at all. Oh, it's a muscle? Is that what that is? Probably. It's called the retractor muscle. Okay, let's get the gill. So we have a heart. It's like this. And then we have like a triangle shape like this. We have little leaflets. It's drawn kind of like an open circulatory system, but apparently it's not. So this esophagus is going to go up here to the stomach. We have a big old cecum. And this is the anus, kind of. Cool, I think we have everything. Funnel, gill. Alright. Oh, wait, ink sac. You need a little ink sac. Okay, good. So now we have like a good drawing of this. Okay, I'm not going to draw this. I feel like this one, the only thing that it was kind of unique was that there's not really a mantle cavity, and then food comes in through these gills and then kind of gets pushed through to the mouth, which is kind of at the back. But so actually, that's maybe interesting is like the hinge part is definitely considered in bivalves like clams. The hinge is the dorsal, but what is anterior? Is it the mouth side or the gill side? Dude, what the hell? Yeah, so the siphons are the posterior. So then okay, fair enough. Yeah. That's actually not a bad drawing. Okay. Um Okay, so what's next is annelids. Okay, so I'm gonna skim annelids and see what's in here. Okay, so they're breaking up into what is it, subphyla or classes or something? I don't know. Oh, here we go. Okay, so this drawing we'll have to do. What is that large tentacle? What is this though? Like, how many tentacles does a cephalopod? Oh wait, and I should draw some eyes. Because I think they have pretty advanced eyes, actually. Gill, heart. Squid. This is like a generic squid. Oh, 8? Oh, okay. 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 So wait, how did we get presented it? We got sort of presented it in sort of an interesting way. So, this is just like a general animals thing. Um, I always forget. I'm almost positive sponges don't do blastulation, right? I feel like it's no. Hmm. Okay, maybe I should write that down somewhere. I don't know. Okay. Let me get some more coffee. Okay. Um, okay, yeah, well, here's what we're doing. We're going through this. These are the lab lectures. Okay. A little bit about development, body symmetry. Yeah, this diagram will definitely need to be added. This is a good diagram because I don't really, honestly, I kind of hate this diagram because I don't even know what this means. Like, how does the coelom like form from this? Particularly the deuterostrum one doesn't really make sense. This diagram's really good. I really like this diagram. This is a great diagram. Okay, yeah, so now we get into circulatory systems. Yep. Yeah, I don't know why I started with Lophotrochozoa. If I'm gonna take notes, I should do it in the right way. So, here's what we'll do is we'll just keep this as the mollusk page, and then I'm gonna go back and do some diagrams of Porifera, because a lot of this I don't remember. I don't know, maybe I should just keep going on. I feel like there's not that much to remember about this stuff. And yeah, I didn't draw this diagram either, but I feel like this diagram's pretty obvious. Besides maybe mesoglia. Myoepithelia. What a great diagram, dude. Come on. Maybe I'll draw this, but I don't know. Okay. Okay, so he goes from like Porifera to Cnidaria, then to Annelida, and then he goes to wait, where's animals 2? Oh wait. Oh, then animals 2. Because we're going into Annelida, where we have to introduce triploblastic and how we get a huge number of extra interesting tissue types. All of this I need to draw. This is going to be intense. All the different types of epithelial tissues. Yeah, oh yeah, okay. Tissue types are going to be... I wonder where in the book it goes into that. Maybe the book just doesn't get into that much detail about. Yeah. So wait, so we're on Animals 2 now. Actually, wait, here's what we can do. We can go duplicate glossary, or not glossary, index. And then we want to look for like connective tissue. Connective tissue. CO. CON. I love that the links are here, bro. That's awesome. Okay, so okay, interesting. So what chapter is this? 40. Okay. All right. So yeah, so we'll get to that. That sounds good. So this is in chapter 40. Okay, this stuff I get. This stuff, not really. Okay, then he goes to. So this was the end of Annelida. This is all Annelida, but there's nothing but that's it. Okay, then we go to Platyhelminthes, which is still okay. So then Platy. This is the lab lecture. Platy. Okay, and then Nematoda. I can turn it on.
Yeah, please. Yeah.
Okay. Okay, so nematodes introduced the first ecdysozoans. Oh wait, we come back to Annelida again. Okay. Holy cow, look at this, dude. Okay, then we do the locomotion thing. Oh, okay, and then we even talk about what is this? Polychaete? Do we even have this? I don't know what the etymology of that is. Okay, yeah, so we sort of just do a review of locomotion. Okay, so E2, this is E1 and then E2. And then after that, E3, I don't even think I opened E3. Oh wait, didn't I? Oh, I did. Then, oh my gosh, dude, how do I fucking turn this off? I hate that. Okay, so then E3 goes, mollusks. Detailed thing about mollusks. Then we go to arthropods. Okay, but then we go to... Let me look at my phylo tree. Because arthropods are ecdysozoans. Wait, let me look at my tree, because I'm confused. Yeah, because echinoderms are non-molting. Okay, fine. So we go from arthropods to deutero echinoderms. And then we talk about its vascular system. Okay, a ton about echinoderms. Just like way too much about echinoderms. like why is there so much about echinoderms? I mean, this is psychotic. Okay. I think my teacher just really likes sea stars. Cause like really, why did we learn this much about that? And then that's where we end off on E3. Then we go to F bugs. Okay, so bugs we go into the 4 classes, the principal classes, 4 classes. Mouth part chewing. Okay, development. That's actually a really short lecture. Okay. It's like 4 pages. Okay, but then my mic's still okay, good. But in this, what does the order of this go? How is this taught? Let's see, we have these open. Oh yeah, this is the diagram, bro. That's the diagram, man. And then this chart. Yeah, there's gonna be a lot of charts to draw here. Because I forget the pressure. But yeah, there's a huge pressure drop across capillaries. Oh, and then this. I'll probably write that down. Okay, I think it's probably good if we just do the overview of each thing. So the lectures, one is going to be circulation. 2 is development and tissues. 3 is feeding. And 4 is digestion. 5 is osmo. 6 is excretion. And 7 is the SE on 5 and 6. Okay, so let's just go through 1. So circulation goes through so first we go from functions, types, components, and blood. So that's circulation. I don't even know how you describe this. Like the cell characteristics, maybe. Extracellular matrix. Fertilization. Cleavage. Okay, cleavage. Deuterostomes, protostomes. Okay. Blastulation. I think we could probably all classify that under morphogenesis. Okay. Okay. So epithelial, connective. Okay. So these are like the. Okay. So tissues. So we can go safely go into tissues. Okay. Tissues. I don't really know why this is in here. When we watch the lecture again, maybe we can try to figure out what he's getting at. What would be the general category of bowel plan and coelom? I feel like that's still morphogenesis. Ooh, actually, wait, hold on. Let me look at this. Annelids have closed circulatory systems? Wow. You know, the way I contextualize most of this is like how complex is the taxa, but I don't have a good... Because arthropods... Annelids are lophotrochozoans and arthropods are ecdysozoans. And in any case, they're both protostomes, and so they both do this thing where these rings of mesoderm. I thought, oh, okay. I thought that they had to be connected, though. I thought there had to be a connection so that it, like, but maybe it doesn't have to be connected. I'm definitely going to draw this. This is a very interesting diagram. Because I feel like I hated... Do we have it in this? I don't think... we don't have the picture that I'm thinking of. But it's probably in Animals 2. This diagram makes no sense to me. Yeah, I think we can probably characterize this under Bauplana. Bauplane symmetry. Seal. Yeah, Coelom. Ooh, that diagram looks interesting. We might have to go to the lectures to get the high res one. Oh, it was down here all along. Okay, so gastrulation is probably the big thing. Is like, how does the gut form? Okay, now we're going back to larval development. I don't even remember him talking about this, dude. I guess it's examples. Maybe I can write examples of development. Yeah, examples. I don't even remember what this is. I don't remember him going into this at all, dude. I think I was zoned out when we talked about this. Okay, lecture 9. Wait. What? No, I forgot one. I skipped over thermo. Okay, so we need to do thermo. Okay, homeo definition. Conform, regulate. Okay. Okay, I would call this considerations or factors. What is homeotherm? Yeah. Okay, homeotherm mechanisms. Okay, feedback. I want to get some of these definite because I remember when I was doing the homework, I didn't really get all the definitions right. Okay, so I would is exchange part of feedback? No, I'll say exchange. Heat exchange. Part-time. Trade-offs. Yeah, trade-offs. Do we have Pokilo as a... We probably do. Oh. Oh, no, because it's poi... Oh, even still. All right. I'm just trying to get a sense of what is in the lectures. Okay, feeding. Um, okay, feeding and digestion. Yeah, I'll just write down why eat. That makes sense. Why eat? Um, Optimal foraging model. Wait. What is he talking about here? Need to maximize benefits while minimizing costs. Yeah, I'll say optimal foraging. Foraging. Specialization. Adaptations and stuff. Suspension feeding. Deposit. Predation. Team. I'll just say team. And then digestion. Okay, I feel like they mostly left. If they come back, I set up another table. Alright, wait right back. Okay. Where were we? I think I'm just gonna move. I hate having to look at. Okay. Tracts. Okay, mastication. Oh my god, dude. The digestion lecture has so much freaking material. Okay, so wait, what is the general category? So we have um okay, mastication. Now, I guess we just go into vertebrate system. Swallowing, peristalsis, gut motility. So in the big list of, okay, I guess we can say motility maybe. Motility.
Or generally and face that they just kind of mop up, and their nectar and to your digestive tract. So we just name enzymes inside and external. So meaning not only outside the cell, but not even where that cell is. Here, to move it through your food process. Rest the larval and the burrow in the sand, and all you see is this big mound of spaghetti at the surface, which then it can retract in. So we have just the to the head, and so that reads going to much the snail here. Some other protrusive.
That's up in a bush. The crab is close. Free. Watch the prey and pull it once. Escape is not an option. Away from here, the prey. Die.
Thing down your mouth into your throat. Mouse. Okay. Now, red moose sleeps.
Wait, is there two
it's just got this extra bone that extends how wide it can stick out. So instead of just the jaw flexing here, it's flexing all the way back here. And then because the two jaw bones, the two mandibles are not.
Oh, I guess this is like. Oh, okay. All right. Yeah, I was like, whoa, this has so much in it, but it's just because it's 2 lectures in one. So, how does he introduce this?
So continuing our discussion of heterotrophy, of acquiring nutrition from resources, organic materials made by other organisms. Last time we talked about acquiring food, capturing food, and ingestion of that food, inserting it into our digestive tract for internal digestion. And so now we're going to see, okay, how does an organism actually get nutrition out of this food that they've just inserted
So he actually
into their digestive tract? And so we'll look at the processes then of this internal digestion. And we're going to start by looking at digestive tracts anatomically. Again, we talked about how these derive from the archenteron of the gastrula embryo, and that particularly with flow-through systems, there are very specific regions of a gut that allow for this sequential processing of food. But typically what we'll see, again, thinking of this as what does a digestive tract need to do? It needs to break down the food and absorb it and distribute those nutrients to the body. And so it's got to do exchange. And so like most exchange organs, we need a big surface area, small distance. And so we look at, for example, a mammalian gut, and we see these ridges on ridges on ridges. So here's the small intestine, for example. We see that the lining of the intestine is wrinkled into these ridges, and these ridges called plicae. And so looking anatomically at the structure of the wall, that there's epithelial tissues. There's smooth muscle, both circular and longitudinally, and then more connective tissue, more epithelial tissue. So all these different tissue types making up an organ. And so the contraction of this contractile muscle and connective tissue wrinkles the surface, giving it lots of surface area. Then if we look at the surface of the wrinkle, we see that the wrinkles are wrinkled. And so these little finger-like projections that we call
feeding
villi.
is like the longest one by far. Feeding is like double the length.
And then if we look closer at a villus, so villi plural, villus singular, so here's one villus.
Okay, all right, so we can keep going then. All right, so we are... here. Okay, we have tracts and sort of a bunch of examples. So we'll say examples. Then we get to okay, mastication. But then we just get to the vertebrate system. So I guess I want to know how he goes from gastric mills to the vertebrate system.
And so forth. Then we need to absorb those monomers into they're also so small storing the food. The gizzard is now this muscular area, usually with hard plates, that then by muscular action is grinding the food up after you've swallowed it. So it's got a hard lining to it, and as well as the animals usually swallow bits of sand or maybe even pebbles depending on the diet of the animal. And that way they have a hard surface that they're grinding the food against. Okay, so it's gastric mastication. Okay, so we need to break it up. So that's physically breaking it. Now if we consider some of the other steps, we'll look at the mammalian system. So in humans, our alimentary canal, our gut now is actually 26 feet long. Right? So since none of us are 26 feet tall, that tells you that your abdominal cavity is like, this is really coiled up quite a bit. And so from the mouth, or the buccal region, we go to the pharynx, the back of the mouth, which in mammals is where your airway separates from your digestive tract. And then from your pharynx, it continues down the esophagus, and so down from your head into your abdomen. And there it hits your gastrointestinal tract, your GI tract, or your gut. And the gut in mammals is separated into a stomach region, a small intestine, and a large intestine. And as we'll discuss, small is the diameter. That it's much longer than the large intestine, but it's smaller in diameter. And then from the large intestines, rectum, anus, body. In addition to the tube, right, the archenteron derived structures directly, we have these glands that feed other material that help digestion happen. So for example, in the head, we have salivary glands. And they've discovered these areas, and since they found an additional set of salivary glands that nobody ever noticed before after centuries of dissecting heads. Then once we go from the stomach into the small intestine, we talked earlier about how the pancreas is there to add more digestive enzymes, but we also have the liver and gallbladder that's adding bile. And so these are accessory organs. So this is the actual gut, and these are accessory organs to the gut. And embryologically, they derive particularly pockets of endoderm off the archenteron. So here's diagrammatically showing the process. So again, from our buccal cavity to our pharynx then to our esophagus, that then goes from our head down, mammals past the diaphragm into the stomach. And so that's one of the things the esophagus has to do, because remember we have that negative pressure breathing going on here. It tends to pull food back up. So you need to have this valve down here that keeps things from being pulled back up. In the stomach, again, since we don't have a crop in mammals, that's our main storage.
Okay. think I'm good. Get like 30 more minutes of sunshine, hopefully. Oh my gosh, what a waste of time.
Structure as well as starting the digestive process. So in birds and insects, that would be two separate organs, right? Your crop and then your stomach and gizzard. Whereas in mammals, is all one organ. Then from there, we go into the duodenum, the first part of the small intestine. And so you can see small intestine refers to the diameter, not the length, but all of this is small intestine. So we're all over the place. And then at that duodenum, where it comes off the stomach, is also where the liver, gallbladder, and pancreas are connecting in. It's right. If you remember, eat that food because we got.
Okay, so it just goes into like the whole thing. You know, mouth, pharynx, esophagus. Okay. Okay, fine. So basically human. So we go into ingest. Then motility. Then processing, I would say. I would just call this processing. Or extraction, maybe. Yeah. Or like maybe we could just say stomach. So like stomach. Intestines. Okay. Then liver and oh yeah, we should just say accessory glands. That's right. Accessory organs. Okay. Then true digestion. This is definitely the most, I would say, detailed. Okay, and now now I guess we get into variation, or system variation, because now we're talking about, you know, how this system. So we take a model system and then we vary it basically, and we say, happens. Now we get okay, that's this. The end of this stuff doesn't really make sense to me, or it's like, whatever. It's all self-explanatory. Okay, that was a tough one. Okay, now osmoregulation. I don't even think I've watched this lecture one time around. Because I think this homework is due oh my fucking god, dude. Pearson is like such a dog shit website. It's hilarious just how bad these education tools are. Like Pearson and Canvas are both total dog shit. Wait, renal function. Okay, so I have 2 homeworks due this weekend. That's great. Oh, I guess because one. What does renal mean? Oh, okay. All right, so I'm gonna leave some space then to talk about Osmo. Oh, dude, come on. Why do they always come back, bro? Fuck. Fuck. Oh, it's raining, bro. It's raining. God. I fucking hate crows, dude. I try to convince myself I like crows, but I hate crows. Fucking crows, bro. I need to get a canopy or something. Just the month of November is insane. It's just like crow. It's like Crow City. Oh, bro. All right, let's just focus. All right, so I think I'm gonna go back to reading the lectures or the Okay. So let's just go back to annelids, because we just got to make progress. This is like, I'm getting pissed off, dude. Can't focus for shit right now. So annelids are ecdysozoans. I think. Wait. No, they're not. Yes, so this is the third in the big categories that we're covering in our class of Lophotrochozoans. Annelida means little rings, referring to the annelid's body resemblance to a series of fused rings. Fuck, dude, look at all this. Oh my god. Where is it? I don't even see it. Oh. Oh, jeez. Okay. Okay, so we'll need to draw that. Oh my fucking god, dude. Where's the where was it? It was Animals 2. That was Animals 3. Do I have this word? I don't even know this word. No, but he definitely does want us to know the order of all these fucking layers. Holy shit, dude. I need to take a walk around the block, dude. I'm on tilt right now. Holy fuck. Okay, to avoid distraction, there's just no other option. We go inside.